In this lesson, we will study meiosis. I strongly suggest that you follow along with your meiosis review assignment, which makes a great meiosis study guide. This assignment is found in Blackboard. By the end of this lesson, you guys should be able to compare and contrast mitosis and meiosis. If you check in Blackboard, below this video there is a link to a handout that has a comparison activity. Be sure to go through this activity. Some of the key differences we're going to see between mitosis and meiosis are how many rounds of division there are and what types of cells we get at the end. During mitosis, there will only be one round of division and we will make exact copies of the original cell. During meiosis, we will go through 2 rounds of division, meiosis I and meiosis II. At the end, we will have 4 daughter cells that are unique, and they will have half the amount of DNA as the original parent cell. So we will take a diploid cell and end with 4 haploid daughter cells. Meiosis is our second type of cell division. This process produces gametes, also known as sex cells, like eggs and sperm cells. During meiosis, cells will divide twice. This is reducing the number of chromosomes by half in the daughter cells. We will produce 4 distinct, very different daughter cells because of these 2 rounds of division. Again, the daughter cells are also known as gametes. Meiosis is used to produce gametes that are part of sexual reproduction. So in sexual reproduction, we have 2 haploid cells, one from a mother, one from a father, and those gametes will combine during fertilization. After fertilization, we have a zygote, or a single-celled baby. That zygote will go through mitosis so that growth can occur, and then we eventually get an embryo and then a baby. So during sexual reproduction, we get contributions of DNA from each parent, mother and father. So each parent contributes 1 chromosome of each type to their offspring. That means that each daughter cell produced during meiosis needs to have half the amount of chromosomes in a parent cell. When we talk about the number of chromosomes in a cell, we are talking about the ploidy. Haploid cells have 1 of each type of chromosome. Below we see an example of a karyotype, which is a picture of chromosomes in a cell, and the cell is haploid because it has 1 of each type of chromosome. Chromosomes are generally numbered with the exception of the X and Y chromosomes. So this cell has 23 types of chromosomes, 1 of each type. Diploid cells have 2 of each type of chromosome. So in the karyotype below, we see that each type of chromosome has 2 distinct chromosomes. So because there are 2 of each type, we say that the cell is diploid. Adult humans are diploids. Humans have 23 types of chromosomes in our cells. Because these cells are diploid, it means they have 2 of each type of chromosome. To get the total number of chromosomes, we must multiply the number of types of chromosomes by the number of each chromosome. This means that an adult cell in a human has a total of 46 chromosomes. Egg and sperm cells are haploid, so these gametes have a total of 23 chromosomes. They only have 1 of each type, meaning they are haploid. When a cell is haploid, it has 1 of each type of chromosome. We can say that N equals the number of types of chromosome. Remember, there's always a 1 in front of that N, or variable. So we say haploids have 1 of each type of chromosome. So to figure out total number of chromosomes, we just have to solve this simple math equation. So 1 times N, we have N equals 3 in this example. So the total number of chromosomes in this organism is 3. In this example, we have a diploid cell. Diploid cells have 2 of each type of chromosome. N still equals the number of types of chromosomes. We have 3 types of chromosomes in this example. 1, 2, 3. Big, small, and curvy. And again, there are 2 of each type in this karyotype. Diploid cells have 2 N total chromosomes. So again, we can solve this short equation using the information here. We know there are 2 of each type. We know that N equals 3. So 2 times 3 equals 6 chromosomes total. You guys should note that in cells from the same organism, a haploid cell will have half as much DNA as a diploid cell. So in this case, our diploid cell had 6 chromosomes. A haploid cell has only 3. Let's practice by looking at this example of a karyotype, or picture of chromosomes. Looking at this karyotype, is this cell haploid or diploid? That's right. This cell is haploid. There is 1 copy of each chromosome for each type of chromosome. Let's review some of the vocab we covered earlier in the chapter. You guys should remember that chromosomes can be made of a single chromatid, and then after replication, we see that the chromosomes have 2 sister chromatids, replicated pieces, that are attached in the middle at the centromere. Now for some new vocabulary. During meiosis, we're going to see chromosome types pair up. These homologous chromosomes are chromosomes of the same type. We know they're the same type because they have many similarities. They're similar in length, they have the same location of their centromere, and they have the same genes on them. So these members of a chromosome pair are going to be attached during the first stages of meiosis. Here's another picture or karyotype of many chromosomes found in a human. We see that they are matched up based on their homologous pairs. Homologous pairs, again, have the same kinds of information on them. We see that this staining pattern is the same on these chromosomes, showing that they have the same genes. They're also the same size and the same shape. There is one exception to this rule, and that is the sex chromosomes. X and Y chromosomes are different sizes. Y chromosomes are teeny tiny, and X chromosomes are very large. Let's get into the process of meiosis. Remember, meiosis has 2 rounds of division. We are going to number these rounds of division, meiosis I and meiosis II. Remember, these are still cells no matter what they produce. Gametes, skin cells, brain cells. So every cell is going to go through interphase. Do you remember when chromosomes and DNA replicate? Right, that's during interphase, specifically the S phase of interphase. Interphase will look the same in every cell no matter what is going on, mitosis or meiosis afterwards. Remember, interphase has 3 steps or stages. G1, where growth occurs and normal everyday functions happen. S phase, where DNA replication occurs. And G2 phase, when the cell prepares for division. A quick tip while you're studying mitosis and meiosis. You guys should draw these processes. If you draw the steps, as I'm going to do in this video, and then write out what actually goes on during those steps, it will help you remember what is going on, and then you'll be able to reproduce it on things like an exam. We've already said that every cell goes through interphase. Remember, during interphase, our cell has its DNA loose and stringy in the form of chromatin. The DNA looks like a bowl of spaghetti at this point. It is not differentiated into distinct X's or chromosomes. The DNA is housed in the nucleus with the plasma membrane on the outside of the cell. Meiosis is occurring in specialized organs called the gonads. In females, that's the ovaries. In males, it's the testes. Remember, the overall goal here is to divide the amount of DNA in half to produce our gamete cells. Meiosis I is going to focus on separating out the homologous pairs of chromosomes. Because there are 2 rounds of meiosis, we have to number the steps of meiosis. For example, the first step of meiosis I is prophase I. During this step, we see that the cells are going to have their nucleus dissolving. The DNA is going to be condensing into nice thick chromosomes that present themselves. Those chromosomes are also going to be pairing up so that these homologous pairs get together. During this step, the centrosomes are also migrating towards the poles of the cell. As they move, they are going to be producing spindle fibers. Once these homologous pairs have attached together, we have a structure called a tetrad. Tetrads have 4 distinct sister chromatids in them. This means there are 2 chromosomes attached and 4 sister chromatids. It's important to note that we get these chromosomes from mom and dad. So one chromosome, or one X, will be from our paternal side. The other side, chromosome or X, will be from our maternal side. Remember, these are chromosomes of the same type. The homologous pairs have attached together. Tetrads are very important so that a process called crossing over can occur. During crossing over, arms of non-sister chromatids cross, pieces of DNA break off and are exchanged. In the end, we have chromosomes from mom, chromosomes from dad, and they each have a piece of opposite DNA on them. This is an important process because it gives us genetic variations in our offspring. This is one of the processes responsible for differences between you and your siblings. The next step in meiosis I is metaphase I. During this step, the tetrads line up along the middle of our cell. This is a random process called random assortment. At this point, our centrosomes are at the poles, and the spindle fibers are going to connect to the center of each of the tetrads. During anaphase I, the tetrads are going to split. We will have one of each chromosome going to each side of the cell. The reason the chromosomes are moving is because the spindle fibers are shortening, dragging them back to the poles of the cell. At the end of this stage, we will not have tetrads anymore. Those homologous pairs will be separated at either end of our cell. Telophase I and cytokinesis happen at the same time, just like in mitosis. We're going to see that cytokinesis is the splitting of the cytoplasm. So a furrow or cell plate will form. In this case, I have a cleavage furrow forming. During telophase I, we'll see the nuclei reforming on either end of the cell. Okay, those chromosomes will start to decondense. And remember, we now have one of each chromosome on either side of the cell. At this time, we now have haploid cells being formed. Okay, count the chromosomes. We see that there is one of each type of chromosome in each side of these new cells. The products of meiosis I is going to be 2 daughter cells that are different and distinct from each other, and they are different from the parent cell. They are haploid cells, having only one copy of each chromosome in each cell. In between the 2 rounds of division in meiosis, there is a phase called interkinesis. This is a brief resting phase. The cells will take a break, kind of catch their breath, and get ready for the second round of meiosis. In Meiosis II, the cells will be focused on separating the sister chromatids and dividing up those replicates into different cells. The first step of Meiosis II is Prophase II. During this step, each of our cells will be going through the same process. We see that the nuclei will be dissolving in each cell. Okay, the chromosomes will again condense into those nice thick X's. And remember, these are haploid cells, one of each type of chromosome. We also see that centrosomes have reformed and they are migrating towards the poles of the cells. As they go, spindle fibers will be formed. The next step of Meiosis II is Metaphase II. During this step, our chromosomes will line up along the middle of the cells. Centrosomes are at the poles, and the spindle fibers will be connecting to the center or the centromere of each chromosome. During Anaphase 2, the sister chromatids are going to be pulled apart. The movement of the sister chromatids is caused by the spindle fibers shortening. The centrosomes are reeling in these chromosome pieces. During telophase 2, a new nucleus will be formed on each side of each of the new cells. The chromosomes on each side will also be decondensing. Remember, these are still haploid cells. They have one copy of each chromosome. We also see a cleavage furrow forming during cytokinesis. These cells will pinch apart into two new cells. Each of the original daughter cells will do this, so at the end of the stage we will have 4 daughter cells. Each will be different and distinct. So, the results of meiosis at the very end of both rounds of division, we have 4 daughter cells produced. These 4 gametes are not like each other. They are not identical to the mother cell either. They have unique combinations of genetic material due to the processes of crossing over and random assortment during meiosis I, metaphase 1. Here's another practice question. The skin cell of a dog has 78 chromosomes total and is diploid. How many chromosomes would be in an egg cell for this dog? Would the egg cell be haploid or diploid? Take a moment to think about this question. So, in the skin cell, this cell is diploid, so it has 2n equals 78 chromosomes total. This means that in dogs, n equals 39. That means dogs have 39 types of chromosomes, way more than humans. We know that the only cells in animals that are haploid are the gametes, which include egg cells and sperm cells. Gametes are haploid, having the n number of chromosomes. That means that an egg cell in a dog would have 39 chromosomes total, and it would be haploid. In summary, this video has covered the process of meiosis. Remember, meiosis produces gametes, which are sex cells used during sexual reproduction. We've gone through the steps of meiosis. Remember, there are 2 rounds, and we must label or number each step in this process. The results of meiosis are 4 distinct and different daughter cells.